Troy Lund is a professor of pediatrics and a bone marrow transplant specialist at the University of Minnesota. And the University of Minnesota, as many of you know, is one of the epicenters of pediatric metabolic transplant in the US. Troy's been involved in many of the advances in ALD transplant strategies and in gene therapy development for ALD, including with the Bluebird therapies. So Troy is going to talk today about some, I think, new thoughts on next steps for ALD gene therapy, and we're excited to hear what you have to say, Troy. Thanks.
All right. Great. Thanks. All right. So thank you for having me. I'm going to, like Josh, I'm going to tell you two tales today. So two different stories. They're quite a bit different, and see what you think of them. So the first tale is called Gene Therapy 2.0, and I'll give you our approach for developing the next version of gene therapy. And then the second story is on anti-MLC1 protein, which some of you are going to already figure out what this is. All right, so what do we need for effective gene therapy? So this is a review we wrote a few years ago. The process is like this. We mobilize stem cells from a patient with GCSF drug over 5 days. We can put that blood in a machine. We can isolate the stem cells. We then expose the cells to a virus. It's a lentivirus. It encodes the normal gene. It pops itself into the genome, and these cells have a normal copy of ABCD1. The patient receives chemotherapy, very similar to a regular transplant. We infuse these cells. The cells work their way to the bone marrow, and there's a small fraction that goes to the brain, and early cerebral disease is arrested. And that process probably takes somewhere between 1 month and 6 months for arrest to happen. All right, so what are the pivotal points for cerebral disease? We need a good virus, one that's non-oncogenic, and we all know the problems with the current virus. I don't need to go into them. We need to infect a lot of cells. When you harvest bone marrow, you are getting billions of cells. They each need to see the virus. The cells need to get in the head, and I will cover all these points. So the problem with the current virus, this is the virus laid out as a piece of DNA from start to end. This is the normal ABCD1 gene. That's not a problem. But driving its expression is a piece of DNA called a promoter. In this case, it's MND is the name of it. It's an old promoter. It happens to be very, very, very strong. If the virus lands in a part of your genome near an oncogene, it can turn that on, and that's what gives rise to cancer. I'm not going to talk anymore about this virus or this promoter. You're in for a real treat because Dr. David Williams is going to speak to you today. He's literally a world expert on designing these viruses and these promoters and the oncogenesis, and it's always a treat when I get to listen to him. Problem number 2, we collect the cells from the patient. We give them the virus. What's lost on a lot of families is that it's a very incomplete infection and repair of the cells. Only about 1 in 5 cells actually are corrected. So when you give this product back to the patient, 75% of those cells are the same defective cells you started with, because the technology is more or less limited to fixing about 25% of them or even 20% of them. And that's just a limitation of using lentiviruses. Problem number 3, we don't talk a lot about, but my lab spent a lot of time on, is that somehow these cells get to the marrow and then they jump into your brain. It's been happening since 1968, which was when the first bone marrow transplant was done, that people engraft their brains. For ALD, we think it's very important and probably contributes to stopping the disease. How it happens, we don't know. How much of those cells get to your brain? We don't know. So there are 2 papers that came out a few years ago. The Lund labs from Seattle actually measured this. They took female recipients who received transplants from males, and those recipients had passed away after transplant roughly about 100 days. They had their brains available to them for research, and they actually could see how many male cells were in the brain, and we'll just call the cells microglia, or microglia-like, or monocytes, or myeloid. They're all the same thing. We don't know exactly what they are, but they counted them. And they come up with an answer of about 4 to 8% of the cells from the donor. That's pretty good. It's lower than I thought. At the same time, my lab was working on the exact same question in mice. And so we published a similar paper the same year, and we counted the amount of brain engraftment in our animal models. Turns out we got almost the identical number of 2 to 8% of the cells in the mice. So it's one of the first times where human and mouse experiments added up to the same thing. So that's the number we're dealing with. Probably not more than 8% of the cells are engrafted into the brain. All right, so what are the solutions to this? We need a better virus. We need to infect a lot more cells, and we need to get more cells into the head. And this is to create the most ideal form of therapy. So the better virus. So my lab simply took out this piece of the gene therapy vector and put in a new one. We're using a myeloid-specific promoter. It's never been tested in gene therapy, but we think it's a good way to go. And we think it'll be much more effective, and we think there's no cancer risk to it. So the first thing we did was we made different permutations of this. Now, scientifically, it gets complicated because you can't just put a virus in and measure ABCD1 protein. You can, but technically it's difficult. The results aren't very clean because that protein's internal. And so we use surrogate markers. And so with this virus, we actually put a cell surface protein on it, known as nerve growth factor receptor. That way we can see how many cells have picked up the virus, just for sake of measurement and ease of measurement. And so we did that. We took human CD34 cells, which are stem cells, added our virus, and then a couple of days later, we looked to see how much expression we were getting. And lo and behold, with our new construct, we can get 75% of the cells infected, which is a pretty high number. That was our target, was about 75 to 80%, so we were pretty satisfied with that. And so now we can infect high numbers of cells. We've altered the culture conditions and added a couple extra compounds to make this happen. We did a zillion experiments, but we went one step further and differentiated these cells. I told you it was the monocytes or myeloid cells or microglia that are in the brain. So you can actually take stem cells and turn them into monocytes in the lab. And it takes about 7 to 14 days. We did that. Monocytes express CD14. And our culture conditions work very well, so we get lots of monocytes in the dish. And we just simply ask the question now, do these cells express the new vector, the new construct? We took out the NGFR sequence, and for scientific reasons, we put in a fluorescent protein. So we mix and match these vectors all the time, depending on the readout we want, but it's all to show proof of concept. And so we were wondering, how many of these cells now are fluorescent? All of them. All of them are fluorescent. So we can now infect a lot of cells, and all the monocytes are expressing ABCD1 from the new virus that we've created. So we thought we're in the right direction. And so there's probably 3 more years of work that I'm not going to go into for the sake of time. But we've transplanted dozens and dozens of mice with all these human cells. They engraft well. They have long-term expression. They express ABCD1. We have biomarkers collected, and some of them are analyzed. And I could go on and on with that. But getting back to problem number 3, which is very interesting to us, is brain engraftment. So we've just submitted this paper on microglial engraftment in our mouse model again. And we did lots of different mouse transplants in different versions. And the paper is really about what are the important things that make your brain engraft in the model. So I'll just give you the conclusion. It's After testing brain engraftment in multiple different mouse models, we conclude that mice that don't repair their DNA have excellent brain engraftment. It's really because they lose their microglia. So to have a high percentage of engraftment, you have to lose what's there. And what's there is your own microglia, which are defective when you have ALD. And so if you can bring those down, then the engrafted cells come up. So point number 3, if you can find compounds that temporarily inhibit DNA repair, you should be able to promote brain engraftment, as simple as that. You can't make somebody genetically deficient in DNA repair because that leads to bad things. But if you had drugs that did it, that would be a good thing because you could just use them temporarily. So are there drugs? Well, sure, there are drugs. They're actually very old drugs, and there's probably 500 papers about this drug called rapamycin. It's also called sirolimus, same drug, just 2 different names. We use it in transplant. We've been using it for years for GVHD and other things. It turns out it actually inhibits DNA repair. It suppresses DNA double-strand break repair. It's been known for a very long time. People have used it actually to try to treat some cancers along with chemotherapy. So I won't go on to how we discovered this drug or why we came up with this idea, but nevertheless, we started working on this. The drug itself is produced by a bacterium, Streptomyces hygroscopius. It's isolated in 1972. It actually was found on Easter Island. The compound's name, rapamycin, is coming from the native name of the island, Rapa Nui, actually. It was initially developed as an antifungal agent. It totally failed as an antifungal agent because it was immunosuppressive, so it made things worse. But as an immunosuppressant, it works fantastic. And we've had it in our drug bank for decades. So the first thing we thought, well, let's test this. Let's actually get some microglia cells. Let's grow them in the lab. Let's measure double-strand breaks, and let's see if the drug actually works. Because we always got to test everything ourselves to make sure it's right. You can measure breaks by using a fluorescent protein called gamma H2X. You can do this under the microscope. These are actually all double-strand breaks within a single cell. You can measure them on the microscope. You can count them. You can use flow cytometry. There are lots of ways to quantify this signal. But the point is, there's a signal to measure. So what did we do? We had control cells. We measured double-strand breaks with H2AX. We added rapamycin at 10 micrograms. We didn't see any effect. Then we added rapamycin at 50, with lots of double-strand breaks. So it is inhibiting DNA repair, as we would have predicted, about 2-fold over baseline. We then exposed cells to radiation. Radiation is a fantastic inducer of double-strand breaks. So when you expose cells to 10 gray, you get all sorts of double-strand breaks. It's like a positive control, because we knew that was going to happen. Interestingly, when you combined rapamycin and radiation, you get more. And when you increase the dose, you even get more. And this is the same if you expose the cells to busulfan. We get almost the identical effect as radiation. So it really doesn't matter, because busulfan induces double-strand breaks as well. So engrafting the brain. So the question is, does this work in mice? So if you give mice rapamycin for 4 days, do their microglia cells go down in number in the head? We really needed to know that before we went on to the next set of experiments. In the lab, we have methods to do this. We can give mice rapamycin for 4 days. We can actually count the number of brain microglia cells they have, and lo and behold, with 4 days of treatment, you can see a substantial reduction of 30 to 40% of microglia cells are gone. Now, whether they'll come back or not after you take them off the drug, I don't know. That wasn't our question. I'd say probably yes, but I'm not entirely sure. We didn't ask that. So now we know that the drug works. It causes increased double-strand breaks. It works in mice to lower the microglia. Does it promote brain engraftment? So now we went to another mouse model where we give black mice rapamycin, plus we treat them with busulfan, about 4 days. We can give them cells from a green mouse. That way you can go back later, a month later, and you can see, well, how many green cells got into the brains of these mice? How many of them became microglia? And did our drug help? Turns out it does. It works very well. So if you look at brain engraftment in the wild type setting, and I'm happy that we actually repeated our own data, it's about 8%. When you put the mice on rapamycin, you can almost double it up to 16%. So now we've found a drug that is in all our pharmacies. It's a safe drug. It's well tolerated. We use it all the time as transplanters. It's repurposed now for another use. And so we've solved yet, I think, another problem with the therapy is that we can get more cells to the head for efficient arresting of cerebral ALD. To see if I have. Yes, so next steps for this is a tip of the iceberg, is to have confirmatory data. We've created every ALD mouse model you could think of that'll take in human cells, NSG, NSGS, NBSGW, quad mice, you name it, we have the mice growing. We're putting together an IND with hopes of a clinical trial within the next 12 to 24 months. So that's that story. That's our take on version 2.0 gene therapy and kind of our approach and how we think about it and what we're planning on doing. So now I'm going to switch gears entirely because the next project is kind of cool, and I always like people's thoughts. And some of you were in the room when I came up with this project, and you'll see what I mean shortly. There's a disease called megaencephalic leukoencephalopathy with subcortical cysts. It's called MLC. It is a leukodystrophy. It's a terrible disease. So I was sitting in Amsterdam with some colleagues. You can see Stefan, Ali. Somebody decided to call this picture the Three Stooges. Thanks. Thank you, Stefan, for texting me this. So we're in a lecture of Mario van der Nepp, who formerly retired. She gives a last lecture, basically, in Amsterdam is the way the system works. It's a very formal process. There's formal dressing to it. And so it was great. It's the first time I've been in something like that. I've known Mario for a long time. She's a fantastic neurologist, discovered single-handedly many diseases. So I'm listening to her talk, and she's summarizing her career, but also summarizing some of her latest research, actually. She touched on several diseases. MLC was one of them. It's a rare genetic condition, affects the brain, it's white matter. The kids have a large head, first year of life, very progressive. They get seizures, stiffness, motor problems, developmental delays, etc. It's caused by mutations in this gene, ABCD1 gene. Of course, it has two names. Everything has two names. It's also known as the modulator of VRAC current 1. So one's the gene, one's the protein. Historically, no cure for this. Sometimes it's also referred to as van der Nepp disease, but there's so many van der Nepp diseases, I can't keep them straight. So these are the MRIs for the kids, so bad white matter. Some of these kids get cysts up here, anterior horn cysts. I've actually seen a couple of kids with this disease in my clinic. For me, they want to talk about transplant, but transplant really doesn't play a role in this disease, and usually what I have to tell families. So, because it's a channel protein, it's not a protein that cells share. This is the protein, the MLC1 or VRAC protein. It sits in the membrane of the cell, and so it has lots of looping fragments that go in the membrane, out of the membrane, in the membrane. This is the outside of the cell. This is the inside of the cell. It has a start and an end. And it's kind of what the protein looks like, and it's important for salt and water balance in the brain. And so, just as I said, it's a protein. It's important for salt and water balance. Yeah, the brain is a very complicated organ. It's made up of lots of different cells. There are astrocytes that send out little tentacles to the blood vessels, and these are little feet that sit in the blood vessel itself. And the feet read the current of the blood. It reads the salt balance, it reads the water balance, and the astrocytes are very important for brain health and regulation of all these salts and waters. There's lots of channel proteins in these astrocyte feet. MLC1 is in green, but there's a whole bunch of other ones, literally probably hundreds. Astrocytes interact with myelin. It keeps your myelin intact. Nice, healthy myelin integrity goes along with healthy astrocytes. So when this is disrupted, when you don't have MLC1 and the protein's broken, everything goes crazy. Everything gets unhealthy. The astrocytes bulge. The myelin is lost. It happens before the first year of life. And these are the MRIs you end up having with the severe myelin problem in this disease. So I'm listening to Mario van der Nap, Dr. van der Nap, talk about this. And then she writes this article last year, Nature Reviews, that multiple sclerosis, which is a disease we talk about a lot because it's similar in some ways to ALD. It has white matter problems, it's inflammatory, there can be gadolinium enhancement. She talked about this and wrote this article as multiple sclerosis, an immune attack on astrocyte-mediated iron and water homeostasis. And what was interesting is she hypothesized that maybe antibodies to MLC1 could be involved with MS, because it's not really clear what the origin of MS is, and it's still a lot of work to be done there. And so what's interesting is that she was talking about this story, and then she finishes her story. And so at the time, this was a question. This was a question she raised to the audience. And this is an example in the pictograph, is that could antibodies come in, disrupt all these channel proteins, and lead to MS and lesion development and disrupt myelin. So she concluded her story because another group just happened to come out with the same research in the same year that said, yes, people with MS do have higher antibodies to MLC1, and it was the first time. So it actually confirmed her idea, and she mentioned this in her talk. It was a very fortuitous kind of timing thing. And this is one of the critical figures is that if you look at the amount of antibodies to MLC1 in control patients, they exist here at 1.5. If you look in MS patients, in fact, they do have high antibodies to MLC1, perhaps as a contributor to MS. Whether it's a disease initiator, I don't know. Whether it just makes things worse, I'm not sure, and I don't think the authors know. But it was a very interesting finding that you could have an immune attack on a channel protein. You can see where this is all going. I hope you can see where it is going at this point. So back to the Stooges. So I'm sitting there with these guys. So Stooge 2, I guess, is me. I was asking myself, well, if this is all true, and I have an interest in ALD and cerebral disease and immunology, could cerebral disease have an autoimmune component involving anti-MLC antibodies? I don't know. This is what I don't. When was that meeting? May? Was it May or? Yeah. Yeah. So May is when we come up with this. So well, let's see what we have. So what we did, we examined boys with cerebral disease for anti-MLC antibodies and boys without cerebral disease. First, we had to make the protein. That took a long time. It wasn't very cheap. So we made the recombinant protein, then we used a technique called Western blot. It's what we do to look for antibodies. And we looked in these boys. So just cut right to the chase, just show you an example. 2 boys, this is their serum on the blot. These boys don't have cerebral disease, A and B. Boy C has bad cerebral disease. And lo and behold, he has a whole bunch of antibodies to MLC1. So we thought, oh, well, that's pretty interesting. Let's screen a bunch more. So as of 2 weeks ago, boys with cerebral disease were at a rate of about 28%. I think we're now at about 40% of the boys that we've looked at. So it's not everybody, but just under half have antibodies to MLC1. If you have ALD but no cerebral disease, 0 out of, I think we're up to about 25 now. So nobody had antibodies if you didn't have cerebral disease. So it's really interesting story. It's not a whole lot more to tell than it was kind of interesting that maybe this plays a role somehow. Yeah, so like with multiple sclerosis, antibodies to MLC1 exist in some boys with cerebral disease. Is there a contribution to disease pathology? That's what we can discuss, and you can give me ideas on what that would be. Future directions, so peptide mapping. I didn't think I'd have this done, but I actually finished it, so I included peptide mapping. There's a technique where you can figure out the part of the protein exactly that the antibody binds to. It's called peptide mapping. You chop the protein up into several hundred little bits. You put them on a chip, and then you can go in with your antibodies, and they stick to the fragments, and then you can read these out fluorescently, and you can see exactly where the antibody sticks to. So we thought, wouldn't it be cool if the antibodies were sticking to the same place in these boys? Because that would really be powerful evidence that it's a true mechanism. So it turns out these are two boys. These are different dilutions of their anti-sera. So it's the same sera in this green, blue, and red, but it's two boys, top and bottom. They are actually sticking in a very, very similar space. This is the peptide. You can see the two overlapping fragments here. So it's really this VDD, SVS, GPH region. That's where the antibody's sticking to. Same place, two different individuals. So we pushed this a little bit further. So this is the outside of the cell, the inside of the cell. Wouldn't it be great if the antibody sticks to a portion of the protein on the outside of the cell? Because that makes a lot more sense. The antibody's floating around and grabs onto the cell and starts an attack. So where is this region of VDD, SVS, GAP? Right there. It's right there in this little loop on the outside of the cell between these two transmembrane regions. So it's a small fragment. It's on the outside of the cell. It all kind of fits together. So that was fortuitous. All this has been crystallized, so you can actually do three-dimensional structures. So I just put this up because it's cool. That's the region it's binding to, is this little box here. These are all the transmembrane regions. You can zoom in. You can put in the specific amino acid. It's just kind of neat stuff. You can dive further. So if you go back in time, we asked the question, has anyone else found this peptide to be immunogenic in completely unrelated systems? And you can actually, there are databases that classify all this stuff. And so the green bars are studies that have been done on the peptides of MLC1. This is what we're looking at here. And in fact, it has been classified. And so lo and behold, years ago, somebody else discovered that this tiny little fragment actually is immunogenic. It can be bound with HLA class 1, B star 1502. So it's amazing that we discovered an antibody that binds to a region on the outside of the cell that somebody also described 20 years ago that is potentially immunogenic. So there you have it. So those are the stories. Always more questions than answers. Anytime I get up there, there's always more questions. What's cause? What's an effect? What's primary? What's secondary? Lots of great questions. And with that, I'll just express my gratitude to ALD Connect and Executive Director Kelly Miattinen. For all the years I've been up on this stage telling you stories. I always try to tell you a new story, a different story, and something that's entertaining and unpublished. So. And this time, Stooges were involved. So that's what I've tried to do. And then thanks to all the patients, families, clinic staff, clinical lab staff, research lab staff, private and public funding agencies that make all of this work possible. Thanks.
Okay, let's see.
Thank you, Troy, for keeping on thinking outside the box in ALE and trying. What do you need to detect these antibodies? Like in terms of, is it a plasma sample? Is it a certain amount? Like the sensitivity of your test, do you have a sense of what you need?
Yeah, it's a plasma sample, and I think we can do it with like 100 microliters of plasma. That's how we've measured these. There are probably other techniques you could do, but that's what we did.
Oh, I'm next. Okay. Hey, Troy. So I'm trying, always just to keep you out of trouble, right? So from the perspective of trying to stay out of trouble, I guess the the world of neuroimmunology, I think, is confounded, maybe infested by spurious correlations between, autoantibodies and certain syndromes. And so I think this is an interesting exercise to do, to really start looking around and seeing how many other antibodies you would likely have also when you have, blood-brain barrier breakdown. So I would just, if I had to take a bet, I would say these are associations that might be more likely secondary rather than is that what you think?
Yeah.
Do I hope? Or
I think that's fair. I think that's fair. It's always hard to know, like, did you develop these antibodies because you had breakdown? The answer is very likely yes. The way to answer it is you'd have to measure the antibodies in boys before they had cerebral disease and then try to get it tied closer together to cerebral disease. We haven't done that. We have a dozen boys or something that we followed every 6 months for a long time, and we have their sera. We just didn't do that. But my guess is it might be secondary or it might be a disease accelerant. We're not sure, but I agree with you. Yeah.
Thank you, Dr. Lund. It's really interesting, and it makes me think of another autoimmune disease that's often misdiagnosed as MS, NMOSD, which also has a water channel that has an autoimmune response, aquaporin 4, in the feet of the astrocyte. So I was wondering, what are your thoughts on that antibody and protein as well, and kind of the research you're doing?
Yeah, I mean, so I've seen NMO for quite a long time, not as many cases as someone like Florian has. Like, I'm sure you've seen a lot of NMO. So I think about it and think about the antibodies. I think when I started doing the work, I thought it was like an analogous process. I think the data is clearer, but correct me if I'm wrong, that the antibodies are causal in NMO. Is that right, Florian and others? Like they're really tied into the disease. And if you get rid of the antibodies, you get rid of the disease. I don't know if that's true for ALD, actually. I don't know if I had said, if we get rid of antibodies, because we've described other antibodies, too, in cerebral disease. I don't know if we got rid of them if the disease would just go away. So, but I'm happy to take other thoughts.
Yeah, great connections you're weaving here, Troy. And I think both the points that Ali and Kate made were important. Mario actually showed years ago when she started this work that there's co-localization of aquaporin 4 to the astrocytic end plates, where MLC also co-localizes to. So I think there's probably a lot longer history of that NMO antibody story. What's curious is that you read MLC and these disorders strike such a different place in the brain than cerebral ALD. And so I agree with you. I think, to Ali's point, this is probably blood-brain barrier disruption, and but whatever you can figure out what might be more specific and in early stages could make all the difference.
Yeah, and Keith and I talked about sterile inflammation yesterday, and that cerebral disease might be a good model of sterile inflammation, as opposed to like having a big bacteria infection in your head. This is inflammation driven by your own body, and all these things are kind of mixed up in that. But I agree with you entirely.
Hey, Troy. Nice work. One question I had about the rapamycin work is rapamycin something you had ever used in cerebral ALD with kind of that analysis of, we did like a year ago, looking at different immunosuppressant agents, just because maybe you're going to get some effect from rapamycin just on the cerebral ALD, either for good or for bad by itself?
Yes. So you and I wrote this paper on different things we've tried. Rapamycin isn't in it because I hadn't. So I just tried it this year, and so I have a boy that's been on rapamycin 6 months now. He has bad cerebral disease. He's getting his follow-up scan next month, and we're going to see the effect of rapamycin. He was too advanced for me to do a transplant, and so I'll be able to tell you in about 30 days what the effect was. And he's been on the drug, good levels for a long time, and no side effects.
Thanks for the talk. Another question on rapamycin. Is the dosage of rapamycin important in the perspective of the possibility of rapamycin inducing cancer? For what I recall, there's like this double hit, like high dose of rapamycin are kind of protective for cancer, and low dosage is kind of increasing the possibility of getting cancer. So I wanted to ask you if this is something that you thought, and if this can hamper your solution for the first problem that you pointed out, like make lentiviral delivery safer in terms of oncological related diseases.
Yeah, this is a good question. So we use doses in the mice similar to humans. So we're on the microgram, nanogram scale. Those are low doses of rapamycin. The cancer risk from rapamycin comes from long-term use over years, so a 5-year use of rapamycin. What we will propose to do is give boys rapamycin for 6 months, and they should have zero cancer risk from that. That's exactly how we use rapamycin in other settings as well, is a defined 6-month treatment plan, and then you're done with it. By that time, it will have had its effect on the brain.
Great. We're a little over time. Maybe one more quick question. Go ahead.
Yeah, very quick question. Thank you, Troy. One thought I had about the transplant questions is we've been wondering how much cross-correction you can get of lipids in the brain. And the transplant experiment is actually a really great opportunity there because you're getting a small fraction of cells, so you're not overburdening with mixed populations and things like that. Are you planning to look at the lipid profiles in the brain of those transplanted mice?
Yeah, Stefan, they're in the box. So yeah, the samples are the brain samples are on the box. I'm going to send to Stefan and look at the amount of cross correction. So yeah, exactly right.
Great, thanks. Thank you, Troy.